Good afternoon, everyone, and welcome to Medical Grand Rounds. Before we begin, I'd like to make a brief land acknowledgement. McGill University is on land which has long served as a site of meeting and exchange amongst Indigenous people. We acknowledge and thank the diverse groups of Indigenous people whose presence marks this territory on which we now gather. Just a reminder to please answer a brief survey that will be sent to you immediately after the presentation. This will let us know that you're present today. And as well, please leave your questions till the end. I ask that you enter them in the chat at the end of the presentation. And finally, our speaker today is courtesy of the Division of Infectious Diseases, and I'd like to ask the Chief of ID, Marcel Baird, to present our speaker. Thank you.
Thank you for that, Nadia. It's a great pleasure to introduce Luke Harrison, who joined our division in September of 2025. So he's been with us for about 6 months since onboarding. Luke's bio tells us he did a PhD at McGill University and omits that he also did a bachelor's at McGill University and then medical school at McGill University, telling us how great McGill University is. And he presumably gets 3 requests for fundraising from McGill Advancement every year. He then went on to McGill University to do medicine, ID, med micro training. And then more recently, he left us. He got sponsored a fellowship by the FRQ, and he went to the Institut Frappier, and he worked with Frédéric Verrier before coming back to work with us. And today he's going to be telling us about something that I'm very curious about and I don't understand myself, which is non-tuberculous mycobacteria. So perhaps he can shed some light for us. So thank you very much, Luke.
Thanks, Marcel, for the introduction. So we'll try to shed a little bit of light, but I think NTM and are sort of the black box of mycobacteria. So we'll try. So thank you again for the introduction and the invite. So today we are going to talk about my favorite group of bacteria, the mycobacteria, and we're going to stay a little bit away from tuberculosis and leprosy and really sort of explore this black box together. So in terms of what I'm going to talk about, we're going to do a little flyover of what are mycobacteria, some of the diversity that exists in this group, and what is the clinical relevance of these organisms in infectious disease. And we'll describe a couple of the presentations. And really this is meant a flavor, a flyover. We're not going to sort of do a deep dive into guidelines or talk about doses of antibiotics. It's more just to appreciate what NTM do to us and where they come from. And then I'm going to segue into talking a bit about diagnostics for NTM, and really with a focus, as sort of a long introduction into some of the work I did in my postdoc, looking at whether we can apply metagenomic sequencing approaches to better understand NTM disease, mycobacteria, and potentially as a modality for diagnostics in the future. So first, just, obviously I appreciate this is a very clinical audience. Let's start with a clinical case. So referred by their patient's rheumatologist, a 36-year-old woman who has productive cough and a normal chest x-ray. She's known for rheumatoid arthritis, which has been well controlled on a JAK inhibitor for 4 years. She's also known for hypertension and as a smoker. She's coming in with a 7-month history of insidious onset of cough that's productive, of sputum with some blood streaking, and she's also quite fatigued. There's no recent travel, and she was born in Canada. In the clinic on exam, she's well. There's some scattered bronchi in the right side of the chest. Otherwise, clinical examination is normal. Labs essentially within normal limits. And from prior to her referral, a sputum culture showed growth of 3+ Haemophilus parainfluenzae. She's referred for CT scan and to investigate the radiographic abnormality on her chest x-ray, and given she's a smoker. And this demonstrates bronchiectasis and atelectasis in the right middle lobe, as well as signs of peribronchial opacities and bronchial mucus plugging. So at that point, spontaneous sputum is collected, and 6 weeks later, all the results are in. And bacterial cultures again grow Haemophilus parainfluenzae, fungal cultures grow some yeast, and mycobacterial cultures grow in 2 out of 3 cultures, Mycobacterium avium with 1+ and 2+ acid fast bacilli seen on smear. And then interestingly, a 3rd culture grows a Nocardia species. Given the Nocardia species and the overall presentation, she's treated initially with regular antibiotics, clavulin, for a prolonged trial of 3 weeks. And repeat cultures are collected, which then again demonstrate in 1 out of 3 cultures acid-fast bacilli on smear and Mycobacterium avium in culture. So this is a diagnosis and a presentation of non-tuberculous mycobacteria pulmonary disease, or NTM-PD, and in this case due to Mycobacterium avium. So these are the kinds of patients that we can see who get sick with NTM. So what are mycobacteria? I thought, I would take the opportunity to sort of step back and say, why do we treat these organisms differently than sort of E. coli, Staph aureus, or more sort of common medically relevant microorganisms? And so the mycobacteria are a family of bacteria in the bacterial phylum of Actinomycetota. So they're a long name, but basically it's a group of bacteria that are mostly gram-positive, include things like Corynebacterium and other actinomycetes, so actinobacteria from the mouth. And these form a distinct family within that group. And really what makes them most distinctive is their cell envelope, or the outside of the bacterial cell. And it's really quite unique in mycobacteria. And they, in fact, have a totally new component of the cell envelope that no other bacteria truly has. And that's known as the mycomembrane. So it's a thick, waxy outer layer that's rich in compounds called mycolic acids, which are not unique to mycobacteria, but have been collaborated within mycobacteria to produce this very characteristic cell envelope. Really, this property has really underpinned their evolution and also explains why these mycobacteria are treated both differently in the clinical microbiology lab and have very different treatment options, et cetera. A lot of the basic biology is driven by this fact. And so just to add a little bit more context to the non-microbiologists in the room, of course, we know about gram-negative and gram-positive. So when we're talking about this, we're talking about the cell envelope of a bacterial cell, diagramized in the corner here. Most clinically relevant bacteria you can sort of broadly group into gram-negatives and gram-positives. And what that really means is they stain differently with gram stain. And well, why do they stain differently with gram stain? And that's due to their fundamental cell envelope structure. So gram-negative organisms are called diderm organisms, or have a double cell envelope that includes 2 membranes, an inner membrane and an outer membrane, and a periplasmic space with a thin layer of peptidoglycan. This is in contrast to gram-positive organisms, which have a single plasmolipid bilayer and then a thick layer of peptidoglycan. And pretty much with some exceptions, almost all medically relevant bacteria can be grouped into these big groups. And so these are things you can think about on a sort of a day-to-day basis as a physician. So E. coli is a gram-negative organism. It stains with safranin in sort of a pinkish color under the microscope, relative to a gram-positive organism that retains a crystal violet from the Grand stain, and that's something like Staphylococcus aureus. And so that brings us to mycobacteria, which are neither and are special, as I've previously mentioned. So rather than having a single plasma bilayer or a double plasma bilayer, they've actually evolved a whole different setup, and this is called a neoderm organism. And so they have essentially the base of a gram-positive cell envelope with a plasma bilayer and peptidoglycan, but then they've reinvented the outer envelope like a gram-negative organism, but in a completely different fashion. And so they stain completely differently. And in the lab, we call them acid-fast because we use a different stain, and they resist decolorization by acid alcohol. And therefore, these are acid-fast organisms, and best exemplified here by Mycobacterium tuberculosis. And so, as I had mentioned, this mycomembrane is really fundamental to their pathophysiology as well as their basic biology. And one thing that this mycomembrane offers is a really thick barrier to moisture loss and a protective layer. This is probably really important in giving mycobacteria resistance to desiccation, as well as resistance to disinfectants and antisepsis measures. From a bacterial perspective, this has allowed them to radiate and colonize a huge number of environments that exist. So pretty much anywhere you look for mycobacteria, you will find them. Freshwater, soils, dusts. The human environment is heavily colonized with mycobacteria, plumbing systems, and a lot of it has to do with their resilience and resistance due to the mycomembrane. This also explains their relative resistance to many sort of conventional, if you will, antimicrobials. So things like clavulin, they're not very effective against mycobacteria for lots of reasons, but a big part of it is the structure of the cell envelope, which explains why these agents are either not able to get in or are just not active because the basic biology is different. So mycobacteria is a pretty diverse group. Right now, there's about 240 known species. So that's species that someone has isolated in culture or sequenced in their genome and given a name to. And this has been classically split into two groups, the tuberculous mycobacteria, so those are the most medically relevant ones. That's obviously M. tuberculosis, the cause of TB, and M. leprae. And then the rest are sort of thrown together as non-tuberculous. But I just want to be clear that this underlies a huge diversity of organisms. And among the NTM, there are organisms that can infect mammals and humans, and there are some that cannot. And so there's really a diversity of both form, function, and environment, as well as ability to cause human disease. So this is just some work from my postdoc where we developed a comprehensive genome database and phylogenetic tree, which is a family tree of the group. And, it's not important to look closely at the names, but I'll just draw your attention to a couple facts. On the outer sort of ring of annotation here, you can classify these organisms based on literature as to whether they cause human disease or not, and whether they're established at causing human disease or they're opportunistic pathogens. And again, it's not to look at the details, but to appreciate that pathogenicity exists throughout the group, and it's not all in one part of the tree. So probably the ability to infect mammals and humans has sort of arisen multiple times in the evolution of mycobacteria. Before we dive back into the NTM, just some brief words. We all know about tuberculosis. I'm not going to spend a lot of time on it. Probably the single most important infectious disease today. Still kills more than 1 million people globally every year. Has infected historical figures and continues to be really a scourge on humanity. So obviously a big important topic, and a lot of research in mycobacteria is on TB and is derived from TB, but will not be the further subject of today's discussion. The other main tuberculous mycobacteria, if you will, is Mycobacterium leprae and lepromatosis, so the agents of leprosy, which, as we know, is a neurotropic disease that causes skin and nerve infection, and interestingly infects both humans and armadillos. So another lot of, we could talk for hours about it, but we're going to continue on to the non-tuberculous mycobacteria. What distinguishes in general, and with any statement in medicine, I have these asterisks, explicit and definitive statements are undoubtedly proven wrong. So in general, M. tuberculosis and M. leprae are host-restricted pathogens. That is, they've evolved to be parasites of their hosts, and they don't live in the soil or the water. This contrasts with NTM, that are primarily environmental organisms. They do live in the water and the soil, and they're happy to live there, but they can also cause disease. Other sort of general characteristics, human exposure to NTM is probably frequent, if not ubiquitous, but disease is not frequent. It's not terribly rare, but it's not every day. And so host factors and a number of other factors are important in that pathophysiology. Human-to-human transmission is probably very rare among non-tuberculous mycobacteria. Again, making definitive statements in medicine is always a bad idea. There's a few exceptions, but those are sort of the general characteristics. And I'll also just take this moment to point out that this 240 species that exists in the described, scientifically known diversity of mycobacteria is very likely a dramatically small part of what truly exists in the environment. So the best evidence comes from, for example, this is a PCR-based soil survey where these investigators took global soils, sequenced a gene that's present in all mycobacteria, and asked, of the sequences we amplified and sequenced, what percentage relate to known mycobacteria? And the answer is about 3 or 4%. So probably there's 96, 97, if not more, percent of the diversity of mycobacteria that is undescribed and undiscovered. But the reason this is likely not terribly clinically relevant is that the ones that tend to infect humans are the ones we find, because the way to end up a named mycobacterium is to be found in a person, for the lab to say, that's not TB and it looks funny, let's do the sequence and let's give it a name. So the known diversity of mycobacteria is filtered primarily through clinical microbiology labs. And so what we have as a known subset is probably at least a good sampling of the things that are biologically capable of causing human disease, or at least to be grown in a clinical lab. In terms of very clinically relevant species, we can sort of, it varies a little bit on geographic region, but if we sort of look at the species that are most likely to cause human disease, they can broadly be categorized into rapid or slow-growing mycobacteria, and that actually sort of correlates with the family tree. If we look at this tree, so each branch on this tree represents a mycobacterial species. All of this sort of group here are mycobacteria that grow rapidly, and that is they form colonies on solid media in the lab in less than 7 days. And all of this other diversity, including Mycobacterium tuberculosis, are slow-growing mycobacteria that take greater than 7 days to grow in culture. But again, If we pass sort of over the more common causes of NTM disease in North America, among the rapid growers, we have Mycobacterium abscessus. These are names that are probably familiar to a number of us. Mycobacteria fortuitum, Chelone, and among the slow-growing bacteria, of course, MTB and lepre, but also Mycobacterium avium and intracellular, or the MAC complex, M. xenopi and Kansasii, M. ulcerans. These are maybe organisms that I'm sure people are more generally familiar with. And just to sort of put those guys on the tree, again, to emphasize that they come from very different parts of the tree. So not all of the things that infect humans are not a closely related group of organisms. The ability to infect humans is distributed among the family tree. And so what do NTM do to us? The main way that humans interact with NTM is through the lungs. So we inhale NTM, and this can cause NTM pulmonary disease, or NTM-PD. And NTM can also cause extrapulmonary disease, and that can either be limited or disseminated. In limited forms are usually skin and soft tissue, lymphadenitis, and there's, any part of the body can theoretically be infected, but these are sort of the main clinical syndromes. So if we just dive in briefly, and again, not trying to do a full review here on these guidelines or this disease entity, but just to get a flavor of the disease as was presented in the initial case, NTM-PD is a chronic pulmonary infection characterized primarily by granulomatous inflammation. In terms of symptomatology, patients present kind of like in our case with cough, with sputum, with dyspnea, some hemoptysis. This can or cannot be associated with systemic signs and symptoms, and can lead to a diversity of radiographic patterns that include nodular, bronchiectatic, and or cavitary disease. So you can see on these cuts from high-resolution CT, you can have sort of more nodular bronchiectatic patterns or more cavitary patterns. And there's pretty recent, quite good guidelines on the diagnosis and treatment of NTM pulmonary disease that's endorsed by both ATS as well as ECCMID and IDSA. So how does NTM pulmonary disease occur? So I told you these organisms are ubiquitous in the environment, and it's true that probably whenever each one of us is having a shower in the morning, you are undoubtedly being showered with some non-tuberculous mycobacteria, whether that's Mycobacterium avium, abscessus. Sort of the fine-grained details of which ones are where is still being worked out, but there undoubtedly is sort of ubiquitous exposure to these organisms. So why do some people get sick and some do not? Well, sort of the easy answer is host factors. I think, for the most part, NTM pulmonary disease is a disease of people that have some intrinsic vulnerability to infection, whether that's a structural problem in the lungs, prior tuberculosis, cystic lesions, chronic lung disease, or some problem with their immune system, whether that's an inherited or acquired immunodeficiency, or sort of relative immunodeficient states that can occur throughout the life. But that's not the only thing, because there are a lot of people walking around with these risk factors who don't have NTM pulmonary disease. And so the feeling is the pathophysiology is multifactorial. It's a combination of host factors. The microbiology does matter. Not every organism is capable of infecting humans. And so some degree of which mycobacteria you're exposed to and at what quantity will also mediate who gets sick and who does not. And there's emerging evidence that perhaps more than just which species of mycobacteria, perhaps there are strains within species that are more virulent or pathogenic to humans. And this is an area of active research. So this is an increasingly important problem. So these are epidemiological data from Ontario, which has some really nice published data. And I'll just draw your attention to this sort of line with the X's. This is TB from all body sites, including pulmonary. And this is pulmonary NTM disease. And so you can see around sort of year 2000, at least in Ontario, the incidence of NTM pulmonary disease now exceeds the incidence of tuberculosis. And this is sort of mirrored in a lot of jurisdictions where epidemiologically tuberculosis is less common. NTM pulmonary disease is becoming more common. And this is just from a recent global systematic review and meta-analysis. And if you look at sort of the slope of the annual rate of change in the incidence of NTM disease, you can see that in most places in the world, it's increasing. Some of this is undoubtedly diagnostics, case recognition, but it may also relate to iatrogenic immunosuppression, increased longevity, comorbidities. It's a very multifactorial thing, but this is an increasingly important problem globally. And I want to emphasize, I'm a microbiologist, so it's fun. It's obviously my inclination to say this, but in the case of NTM-PD, it does matter what the microbiology is. So this is not a a sort of monolithic disease entity where you can just give the same antimicrobials and patients, do better or worse. The organism that causes the NTM-PD is a very important predictor of both clinical outcomes. So on the right here is also from Ontario epidemiological data showing that overall survival from the NTM pulmonary disease infections does actually segregate depending on the infecting organism. If I just, on the left here, again, this is not to review guideline level sort of data comprehensively, but just to appreciate that the different organisms that can cause NTM pulmonary disease have very different treatments. So if you have a patient infected with Mycobacterium abscessus, you're going to be giving a macrolide, and then you may be considering maybe even a beta-lactam, cefoxitin, linezolid, tigecycline. But if you have a patient who's infected with Mycobacterium avium, completely different treatment, although you still give us a macrolide. Now we're talking about ethambutol and rifamycin, so more TB-like treatment. And then some of the other organisms, again, have quite sort of idiosyncratic drug susceptibility and management recommendations. So the point here is that the microbiology of these diseases matters. Just a brief flyover of extrapulmonary non-tuberculous mycobacterial disease. We can broadly sort of classify that as disseminated or non-disseminated. Disseminated disease is really generally a disease of profoundly immunocompromised patients. And so this is best known from the AIDS epidemic in the late 90s, or in the 90s, 80s and 90s, where disseminated disease from Mycobacterium avium intracellular, or MAC, was really a profound feature of end-stage AIDS. And I just think it's really interesting to go back to some of the data from that time. This is just a case series from San Francisco, where they looked at the percentage of patients who ended up having mycobacteremia with Mycobacterium avium over time. And, it's a little bit of a selected series where they were sort of identified by the fact that a clinician had sent a mycobacterial blood culture. So there is some it's not a sort of all comers. You can see the percentage is really quite profound. So I think this speaks to the ubiquity of the organism in the environment, that if the host is vulnerable enough, there is sort of ongoing exposure that in the case of advanced HIV/AIDS with a low CD4 count, you're not sort of doomed to necessarily have MAC in the blood, but it's certainly a pretty good probability. And this is a much smaller problem overall than NTM pulmonary disease. So again, from Ontario epidemiological data from the 2000s, we're talking sort of total case counts less than 100 for most of these syndromes. So really, seen in very immunocompromised patients. And again, coming back to some 1990s AIDS data, again, from the West Coast of the United States, this is a really interesting study where they looked at initial mycobacterial blood cultures that were positive with Mycobacterium avium or intracellular. And then they looked it was a prospective cohort, and they looked sort of before and after, where else were these organisms found in these patients. And so what they showed in this study was that before the index blood culture with mycobacteremia, you could find Mycobacterium avium in either the stool or the lungs of these patients. So probably these were patients who were acquiring infection from the environment, either through the lungs or more likely through the intestinal route, sort of drinking M. avium in the water, it infecting the gut, and then disseminating in the context of profound host vulnerability. I think I'll have time to do a quick aside, just because I think it's a really interesting organism, just to sort of underlie the diversity within the mycobacteria. So not all NTM are just, a different name for a bug that causes, cavitary lesions in the lungs. So one particularly interesting case study is Mycobacterium ulcerans. So this is the agent of Buruli ulcer, which is a neglected tropical disease. This organism is very closely related to a different mycobacteria that's very common in marine environments and fish, called Mycobacterium marinum. What distinguishes is the presence of a large plasmid, so that's a circular piece of DNA, that contains the genetic machinery to produce a very important toxin called mycolactone. And mycolactone is essentially a cytotoxic molecule. And when this organism becomes inoculated in the skin and it produces mycolactone, it causes disruption of nervous signals, so it becomes asensate and causes extensive tissue necrosis. And so you get these horrific wounds related to Mycobacterium ulcerans. For a long time, this was thought to be an organism that lived like its counterpart Mycobacterium marinum in water, and people would be exposed by touching water and having a cut and the bacterium being inoculated. But most recently, through some pretty elegant experiments in the distribution range, particularly in Australia, so you can see it's sort of distributed globally, but with a very particular geographic distribution. But recent work in Australia has really demonstrated that, in fact, Mycobacterium ulcerans and Buruli ulcer, at least as it exists in Australia, is actually a vector-borne disease from mosquitoes. And so there's an interesting life cycle between infection of possums, mosquito bites, and humans being inoculated with M. ulcerans through vectors. And whether that's true in Africa is not so clear, but just a fascinating data point in sort of the diversity of mycobacteria. Okay, so that's sort of a very fast flyover of some of the diseases that NTM cause. And now I'm going to jump into diagnosis of mycobacterial infections and then some of the work that I've done looking at sort of metagenomics in that context. So first, I thought, when you send a culture to the lab, sometimes it's a bit of a black box for the non-laboratory physicians among us. So I thought I would just go over what happens when you send a mycobacterial culture to the lab, and how long it takes and why it takes a long time, and then what are some of the things that we can maybe do to speed that up. So in terms of diagnosing a mycobacterial infection, if we're talking, let's just keep it simple and talk about pulmonary disease. You have your patient spit into a sputum sample into a cup. You send it to the microbiology lab, which in the MUHC is at the Royal Victoria and the Research Institute in E5. The first thing that'll happen is a technologist will look at it under the microscope and perform what's called an acid-fast smear with our stains. And if they see mycobacteria or acid-fast bacilli, we'll then perform a direct PCR using a commercial PCR kit to look for TB. If that PCR test is positive, we say this is presumed TB, and then it's confirmed with culture. If it is negative in the presence of a positive smear, this may be an NTM, maybe TB, needs confirmation. At the same time, it is inoculated to a liquid culture system called MGill, where the sort of sputum is inoculated into these liquid culture vials, and it's put into an incubator. Then, at some number of weeks after incubation, and again, there's a lot of variability depending on the organism we're thinking about, but these slow-growing mycobacteria, after inoculation, if there's a low bacterial load in the specimen, this can take 3, 4, 5 weeks to grow in culture. And the culture system is sort of automated. There's a fluorescence when growth detected, and it sort of rings a bell. Your culture is positive. Then this is the subcultured and also examined again under microscopy. We look for acid-fast bacilli, and a PCR is again performed, this time not on the specimen, but on the cultured organism. If that's positive, okay, we confirm TB, and it's sent to our reference lab for drug susceptibility testing. If it's negative, again, this is a presumed non-tuberculous mycobacterial infection, and it is again sent to the reference lab for identification and drug susceptibility testing. So in Quebec, this is done at the LSPQ in the West Island, and they will receive the specimen. They will then reculture it on both liquid and solid media, and then once they have nice colonies, they will identify this organism. At least at the LSPQ, it's done by sequencing of a specific gene that's common to all mycobacteria, the 16S rRNA. And then they will do drug susceptibility testing by broth microdilution. I'm speaking specifically about NTM. And so that takes another couple of weeks. So if you sort of stack it up and then you include the delays that are inherent in specimen transport, result reporting, which, to be honest, at the moment, until the provincial laboratory information system is implemented, this is faxing back and forth. We're talking about a pretty significant delay to go from sputum to telling you that it's Mycobacterium avium that's susceptible to these antimycobacterial drugs. Another aspect of diagnostics and culture-based diagnostics is interpreting the meaning of a positive culture. So I think I've showed you that NTM can certainly cause important disease in humans. They are ubiquitous in the environment, and the problem with things that are ubiquitous in the environment is they have a way of making it into clinical specimens. And so they're considered ubiquitous, and they can be also potentially transient colonizers, although if I had more time, we could talk more about the evidence that supports that hypothesis. What do we do when there's a positive culture? If it's a sterile site, if we're talking about blood culture, or we're talking about a wound that has purulence, and tissue culture has been sent and it grows a pathogenic mycobacterium, well, you've made the diagnosis. In terms of pulmonary disease, there are specific diagnostic criteria that help us interpret a positive mycobacterial culture. So in order to be diagnosed with NTM-PD, according to the guidelines, you need to have both clinical criteria, radiographic pattern, and microbiologic criteria, which in the current guidelines is at least 2 different sputum samples that are positive for the same organism, or 1 BAL, or there's some sort of another criteria based on histopathology and culture. And I don't want to spend too much time reviewing the guidelines, but the point being that a single positive culture requires further evidence in order to understand its significance. And just to underlie that, these are data actually from this institution that was published by Dick Menzies and, well, a number of people in this room, and some of the recent NTM-PD fellows. They looked at retrospectively non-tuberculous mycobacterial cultures here in this institution and looked at whether patients, to look at many things, but among the things they looked at were whether patients met ATSA or IDSA criteria. And so if you key off the culture, and they looked at some 400 specimens, only 68 of those 400 actually met the clinical criteria. So we're finding a lot more mycobacteria than are necessarily clinically relevant. So what are some of the other what are some of the limitations of these culture-based diagnostics? So one, I think I've already impressed upon you, there's some delays that are involved, particularly for slow-growing mycobacteria. And, sort of also interesting is to how much culture itself, the diagnostic tests, is a filter for which mycobacteria we actually find. And is this clinically relevant? So there is a notion that mycobacteria grow differently in our culture systems, which are adapted to find things that are sort of grow in humans. We incubate at 37 degrees. So how much is what the lab reports out of a patient's sputum reflect the disease process in that patient? Are we capturing all the species that are present in that patient? Is there an important heterogeneity in strains of the same species that exist at the time of diagnosis? Is there perhaps heteroresistance to antimicrobacterial drugs that exists in the patient, but when we put it in the culture in the lab, the strain that happens to grow in our cultures maybe is susceptible to an antibiotic? The patient may harbor populations that are both susceptible and resistant. Now, what about mycobacteria that don't grow so well in our particular culture system? Fastidious or unculturable mycobacteria. And there's some emerging evidence that in NTM-PD, we actually do see a lot of diversity of strains and even species in the same person through infection. So this is a recent study in Chest in 2025 in South Korea, where they were able to show that even if you keep isolating Mycobacterium avium throughout treatment of NTM pulmonary disease, it's not necessarily the same Mycobacterium avium that you had at the start. In this case, they didn't use whole genome sequencing, but the principle is the same, that culture may be acting as a filter, and the diversity of infecting strains and species may be more pronounced than we understand. So this will allow me to segue into sort of one of the things I'm quite interested in is how we can use metagenomics to sort of start exploring these questions. And I want to be clear that this is pretty far from a diagnostic test that we can go out and order from a company. There's a promise that we can use perhaps metagenomic sequencing, and I'm going to sort of give a primer on metagenomic sequencing, what that is. But if we focus on what it could offer for mycobacteria, so this is the concept of taking the patient's specimens, sequencing all the DNA that's present, what can that tell us about the mycobacteria that are infecting that patient? So the first thing it could say is, can we use the sequencing of patient specimens for rapid identification of the mycobacteria that are infecting in the sputum? Can we say something about strains, different species that are co-infecting? And can we even potentially derive genotypic drug resistance information from that metagenomic sequencing? And further, sort of away from the clinical side, kind of more in a biology question, metagenomic methods are also offering promise to better delineate the habitats of mycobacteria. So I had said, and if you read any mycobacteria paper about NTM, the first thing they'll say is mycobacteria are ubiquitous organisms that live in dust, water, soils, and they provide about 4 citations. Now, that's true in those citations that someone looked at some dust and found mycobacteria. But in a broader sense, these niches are not so well defined. So is Mycobacterium avium, yes, you can find it in water, but is it in ponds of a certain pH? Is it in water distribution systems of a certain pipe type? There's probably very important delineations in the environment that mycobacteria live that we can start getting at with metagenomics. And that may in turn inform our understanding of how people are exposed and infected by mycobacteria, which may in turn potentially down the road offer us some guidance as how to avoid infection. And so really the question is, can you use metagenomics to go from several weeks to go to several days? Can we skip sort of all those intermediate steps, take the sputum, do some DNA sequencing, find the species, and maybe provide some information about drug susceptibility? So that's the promise. And to be clear, we're a long way from there. And I'm going to talk a bit more about metagenomics, and I appreciate that most are probably sort of most familiar with metagenomics as it relates to the microbiome. And there's just, an explosion of research over the last 20 years looking at how the microbiome is influencing multiple aspects of human health and disease. This is just from a recent review of microbiome and cancer. Just sort of like these are all citations in journals, just sort of a dramatically large amount of literature looking at the influence of microbiome in general on human health and disease. That is not what I'm going to be talking about today, but it is the same methods that underlie those studies. So let's talk a little bit about metagenomics. So what is metagenomics? So metagenomics is the study of all genomes present in a given sample. And you can kind of broadly split metagenomics into two big categories. You can talk about targeted sequencing or targeted next generation sequencing or amplicon sequencing, where you take interest in a specific set of genes and you perform targeted amplification of those genes. And those genes may exist in multiple organisms, so it's still metagenomic in the sense that you're amplifying genes from multiple organisms. But it makes things a bit simpler when you're dealing with the amplification of a single gene and then asking how does the different sequences that I recovered in my specimen relate to some database of those genes. What I'm more interested in, and what I think the field is sort of going to in the longer term, is untargeted or shotgun metagenomics. And that's so where you don't focus on a specific gene, you're just interested in understanding what is the total DNA that is present in a given sample. And just before I move on, just to say that amplicon sequencing and targeted next generation sequencing is a thing for mycobacteria. To my knowledge, it has never been applied to diagnostics in NTM outside of a single study in China using something called the TB Pro assay. But if you sort of Google that, you don't get any English language results. So still, there is some things that exist, but don't have time to talk about that. I'm going to focus more on shotgun metagenomic methods. And I really want to sort of everyone to understand what I'm talking about. So I think I was going to take the time to go through very quickly what exactly is metagenomics in a sort of a nuts and bolts type approach, and how do we interpret the data? So first, let's just step back one further step and ask, how do we sequence a bacterium? So if we grow MTB, we have MTB in a sputum, we grow it on a plate, we have some bacterial cells. How do we go from that to whole genome sequencing? And then it'll be sort of become more clear how that relates to metagenomic sequencing. So you take those bacterial cells, you lyse them or break them apart, you perform a number of sort of chemical steps in the lab to extract the DNA, which you then purify, and then you can sequence. So what does it mean to sequence when we're talking about next generation sequencing? Not going to dive too deep, but just to have an idea, we create a sequencing library by taking the DNA that you extracted from your bacterial colony. You sort of break it up into fragments. Those fragments get little adapters ligated to them, and this is sort of Illumina technology, but there are similar and different technologies from other vendors. Those adapters allow you to fix the short pieces of DNA fragments onto some device, in this case a flow cell. There's an amplification step to amplify those fragments, and then through reversible chain termination and laser light, the details, again, not super important, but you're able to sequence those fragments and derive the ATCG, the DNA sequence of the fragments present in that specimen. And so what do you end up with? You end up with a set of DNA sequencing reads. So you could have millions of these reads, but they're short. In this case, for Illumina technology, somewhere between 100 and 350 base pairs. And so then how do you say something about those reads? Now, in the simplest way, you can say, well, I've sequenced TB, so I want to understand what those reads say about the genome of the TB in my specimen. So you can take, for example, a reference genome, you can sort of linearize that in some software, and then we can sort of find where those individual sequencing reads match against the reference genome, and then ask how well do they match. So again, not going to be too pedantic, but what you're seeing here is the reference genome here, and then each of these represents a sequencing read. And where they're gray and there's no color and there's no nucleotide indicated, it's because every nucleotide here is essentially the same as the reference. There may be some sequencing errors, but they're hidden here. But in some positions, where in the reference genome here, there's a G, all of the sequencing reads have a T. And so that's how you derive information from the sequencing reads to say, my genome of interest is a T in this position instead of a G in the reference genome. Okay. So that's whole genome sequencing with short read data. Now, what about metagenomics? So how does that differ? So in metagenomics, you take a complex community. In this case, we're talking about multiple microorganisms. You do the same thing, you extract all their DNA, but it's all sort of mixed together. And then you do metagenomic sequencing where you do the same procedure, but in this case, rather than having reads that come from a single genome, now you have reads that come from multiple different species and organisms all jumbled together. And so this becomes a much more difficult computational problem to say something about what those reads mean. And if we focus on sort of clinical aspects, so doing this in a human, clinical metagenomic sequencing will therefore give you a set of sequencing reads from a human specimen that will represent a few things. If you're in a sterile site, this will represent sequencing reads that come from the human, us, the host, the pathogens, if this is an infectious disease and you're trying to diagnose it, and then some contaminant organisms. So, a sterile site might be CSF or blood. And if you're interested in a non-sterile site, which you might be in, for example, non-tuberculous mycobacterial pulmonary disease, you're going to get DNA from the host. You're going to get reads from the pathogens, let's say the NTM. There might be some lab or environmental contaminants. And then there may be all the messiness that exists in human lungs. So there's going to be bacteria, there's going to be fungi, there's going to be candida, and there's going to be viruses. There's going to be really reads that are derived from multiple organisms. So this is a much more complicated problem. So how do you deconvolute all those jumbled together reads in the metagenomic data set? There's really kind of two broad strokes approaches. One, you can take a computer algorithm and you can look for overlaps between those reads. Find regions where those reads overlap, and then sort of stack those. And if you have enough of them, you can stack them and build a longer and longer, what we call a contig, so an assembled piece of DNA. And this is called a metagenome assembled genome. So you can sort of take those reads, stack them together, find the overlaps, and build a DNA sequence. And you can sort of look at what that genome, you can compare it to other genomes using a number of bioinformatic approaches and say something about the organisms that are present in your sample. What's more common in clinical metagenomics when the organism is perhaps present at not an overwhelming abundance is to do what's called reference-based genome alignment, or reference-based read profiling. And you take this jumbled reads and you compare it to some database and you ask not what are the overlaps between my reads, allowing me to build a longer DNA sequence. It's more which read came from which organism based on a reference database. And that's really the approach that most clinical metagenomics work has taken, although not exclusively. This is not sort of theoretical. There are people that are doing this. This is data from Charles Chu lab at UCSF, in fact, published by Patrick Benoit, who's now on staff at CHUM in infectious disease and microbiology. And they looked at retrospective over 7 years of offering clinical metagenomic sequencing, which is based on read-based profiling of CNS infections. Again, we could talk for an hour about this one paper, but what they demonstrated is that it's pretty good. You can sort of, overall comparing to all the other microbiologic ways to diagnose CNS infection, you can have a sensitivity of about 63% and a specificity of about almost 100%. Some of the sensitivity is lost just because there is no nucleic acids for some infectious diseases and CSF infections. There's some nuance there. It works pretty well. So you can deconvolute those reads, you can remove the human, and you can say something about infecting microorganisms. It's not perfect, and there are some organisms, so these are the ones that they sort of missed, that were diagnosed by other modalities, and you can see MTBs in there too. So it's not perfect, but there's a proof of concept there that this can be offered as a clinical test in a sterile site. We're interested in mycobacterial infections and saying something about sputum, which is a much more complicated specimen, a lot more messiness. And so what are the challenges to applying these sorts of methods to NTM diagnostics? Well, there's really a lot of challenges. The first is that NTM are probably not present at overwhelming abundance. So when the patient coughs up the sputum, yes, there's NTM in there, but there's probably a relatively small amount of NTM genomic DNA relative to the human and other and potentially other respiratory organisms. And also potentially, based on what I told you, multiple strains and multiple species of NTM present. So that makes the computational figuring out which one is present much more complicated. And I told you that species matter, and some of these species are actually pretty closely related in the mycobacterial family tree, so that makes it even more difficult to tell them apart. And finally, because we've not really done this before, how do we interpret a positive? Is it normal to find NTM DNA in a healthy human sputum? No one's really asked that question. And without going too deep into the weeds of the bioinformatics, one of the main challenges that I brought up was if you have multiple mycobacteria in one specimen and their genomes are very closely related, the reads that you sequence from them are also very closely related. So how do you use computational methods to say which species is truly present? And just to give an example, mycobacteria exchange DNA in the environment. So if you have two species of mycobacteria swapping DNA, you may have reads that came from the genome of one that match the reference genome of another. And so it's really important. There's a number of methods that have been developed that appreciate this problem and try to look at multiple metrics of where the reads align in the genome, how closely they align to the genome, to try to really reconstruct which species are present. And so one of the projects I did during my postdoc at Amalfi was to look at benchmarking how well these methods are at figuring out which mycobacteria are present in a complicated metagenomic sample when you have multiple mycobacteria. And so to do this, we did some in silico simulations. The simulation procedure was complicated, not enough time to go through it, but we basically simulated the presence of multiple mycobacteria in a complicated metagenomic sample. And then we took some of the existing pipelines and methodologies that exist and asked how well are they able to tell us which mycobacteria that we know are there are in this specimen. And this was work that was done with a PhD and a master's student in Fred Villier's lab, Nyere and Juba, and so thanks to them. And we looked at our precision recall curves. This is essentially the same as looking at a ROC curve of sensitivity and specificity. And we compared different methods. And if we look at a method that's kind of similar to what the Chu lab does with sterile CSF samples, they use essentially a read alignment count-based approach. And so in our benchmarking, that's not the most precise approach. For a sterile site, it's probably okay. But when you're dealing with complicated metagenomes where you have multiple organisms of interest present that share overlapping genomes, you can really get a lot of problems deconvoluting the reads. And I'll also draw your attention, interestingly, to probably one of the most widely used metagenomic profiling software suites called Kraken. Also has decent performance, but not the best. Recently, in the last year, we have two new programs, one called MetaPresence and one called Self. If I had more time, I'd tell you about how they work, but suffice it to say that they really perform very well. So they're much better at using all the information that's contained in those sequencing reads to reconstruct which mycobacteria are present in a given specimen. And we can look at how those methods also performed in terms of their ability to the sort of lower limit of detection, how few reads you require. And we can also look at their ability to discriminate between the species of mycobacteria truly present in a sample when you throw in a bunch of confounding reads that either come from the human genome, from closely related non-mycobacterial species, drawn from two different databases. And again, if I had more time, we could talk. I'd be happy to chat more about what this shows. But to sort of sum it up, this new method, MetaPresence and SILF, are really much more precise. So they make much fewer mistakes deconvoluting these complicated metagenomes where you have multiple mycobacteria present, and they're able to say, the mycobacteria that are truly present are X, Y, and Z. And this differs pretty dramatically from some older methods, like, for example, Kraken, which when you're interested at the species level really does not perform very well. So I had introduced a further challenge, which is interpretation of something like this is contingent on our understanding of what's normal. And so no one has really sort of looked carefully for what's present in a healthy human's microbiome. Are mycobacteria actually present in all of us? Or are they sort of ubiquitous in the soil, dust, and water? Or is there a component of a normal, healthy human mycobacterium? So we took the best performing methods from our in silico benchmarking study and applied it to what's really the gold standard in terms of microbiome studies, the Human Microbiome Project, and looking at samples from the oral cavity, stool, vaginal specimens, skin, and the nose. And, we find some genomes of mycobacteria, but we find none in the stool, none in the oral cavity, a couple in the nose, although for the most part, M. gordoni is considered a potential laboratory contaminant. So this is of sort of uncertain significance. And some on the skin, which sort of goes with, there's dust in the skin, there's dust in the environment, less clear. So we got initially a little excited when we found 7 identical species of Mycobacterium coloniae in vaginal microbiome samples. And then we looked very carefully at how these specimens were collected, and clinicians were allowed to wash the speculum with tap water before they performed these examinations. And so Mycobacterium coloniae is one of these classic found in water distribution system organisms. So we think this is most likely, an artifact of just this collection procedure. So all that to say, based on the human microbiome data, probably not a component of a healthy human mycobacterium. But then you'd ask the question, there's a sort of important part missing, which is what's going on in the lungs. The Human Microbiome Project, which is sort of the gold standard initial characterization of the human microbiome, notably does not include lung specimens because of their low biomass and difficulty working with them. So it was not a specimen that was targeted initially. We do have some data, and I have to say, looking very carefully, there's really only a couple of studies where healthy humans gave sputum samples and shotgun metagenomic sequencing was done. And probably the best one was published in ATS journal, and they had 26 specimens of healthy sputum, as well as some disease counterparts with shotgun metagenomic sequencing. So if we apply again our validated benchmarked methods to detect mycobacteria, we can find no mycobacteria in the sputum of healthy lungs, and some percentage of, in this particular cohort, and I expect this would be very cohort dependent, we do find some mycobacteria in sort of structurally abnormal lungs, which is, I think, in line with expectations. And certainly some of them are organisms that have clinical relevance, for example, Mycobacterium abscessus or colonae. So this sort of, again, probably not good evidence that there's a healthy human mycobacterium, which allows us to better contextualize the results if we apply this to real sputum specimens. So where to go from here? So I'm interested in building on these benchmarking studies and initial work with sort of looking for the human mycobacterium, but adapting this pipeline and benchmarking studies to Oxford nanopore-based sequencing, which offers much longer reads, which offers more specificity. And if I had more time, I'd be happy to chat about why we think that's a good idea. Looking at in silico benchmarking of the feasibility of drug resistance prediction from sputum using metagenomics and seeing if we can define a lower limit of detection for that kind of assay. And of course, finally moving into actually sequencing and smear-positive NTM sputum with nanopore technology and really testing these methods in the real world on clinical specimens. So that's what I wanted to say today. Again, I'd be happy to take questions and discuss. And thank you to funding sources.
All right, I'm going to look at the chat here. Do we have any questions in the chat? We will start at 12. Wilson, Marco Ad, read it in. Can you have a negative PCR and a positive culture and or vice versa from Sasha?
So definitely, most of the literature would come from Mycobacterium tuberculosis, but if we're talking about direct PCR on sputum, culture definitely has a greater sensitivity than direct PCR on sputum.
Dr. Doss has asked, apart from Mycobacterium tuberculosis and Mycobacterium ulcerans, do other mycobacteria produce toxins?
So they definitely produce, the mycobacteria in general can definitely produce secreted substances that are important in their pathophysiology and are virulence factors. In terms of potent toxins, I think mycolactone is the most well described. And in fact, the plasmid mycolactone, I sort of glossed over some of the diversity. The genetic machinery to produce mycolactone actually exists in multiple forms and in multiple related species of NTM that can cause similar disease.
And Dr. Doss also asked about phages. Are there phages in the NTM?
Yes. So there's a whole, so like mycobacteria are a distinct group of bacteria, they also have their distinct group of bacteriophages, which are called mycobacterial phages. So yes, there are. And yes, you can see, yes, you can see phage in DNA sequencing. Yes, you can find traces of prophages.
We have a question from the room, Mark.
Just in terms of the incidence of clinical disease, you mentioned 100 cases in Ontario. I presume that's the entire province of Ontario. And how are clinical services set up for non-TB bacteria in the province? Is it distributed amongst the academic centers? It's centralized here, and what's optimal?
So the question for the Zoom people is, if there's 100 cases in all of Ontario, how are the medical services distributed?
So just to be clear, I think perhaps I was, let me just go back to the slide. So that was referring to disseminated and extrapulmonary disease. So for extrapulmonary disease, Yeah. So for pulmonary disease, there's a significantly higher burden. In terms of the clinical, so
It's 10 per 100,000.
Yeah.
So you can multiply that by 16.
So for
160.
diagnostics in Ontario, all mycobacterial cultures, as far as I'm aware, are done in the Public Health Ontario lab. So they have quite a centralization of mycobacterial diagnostic services. In terms of their care pathways, I know there's the UHN has an NTM clinic. As to how it happens outside of the UHN, I'm less familiar in Ontario.
And what about Quebec?
So in Quebec, most larger hospitals will have a clinical mycobacteriology lab, but all NTM identification in Quebec is done at the provincial reference lab, which is the LSPQ.
Dr. Flagel, and then there's another one in Zoom from Sasha.
Among acid-fast organisms, what factor or characteristic determines whether they're transmissible by aerosol?
That is an excellent question.
Yeah.
There's some debate as to, so there's some work in Mycobacterium abscessus about the aerosolizability of that organism. I don't know, I think and certainly in TB, there's research on some desiccation resistance aerosolization. Outside of those organisms and avium, I don't think there's much available, but I'm happy to be corrected.
Sasha wanted to know if hand sanitizers work.
So for Mycobacterium abscessus, it is resistant to alcohols. So I don't know if that's ever been tested clinically. In general, a lot of our products, like the wipes that we use in the hospital, are tuberculocidal, so are expected to work against mycobacterial, but they are generally resistant to alcohol disinfection.
And Dr. Rosenblatt wants to know if you envision the molecular technologies replacing the functional assays as is happening in genetics.
This is a really good question. I guess I'm a microbiologist. As much as I enjoy bioinformatics and DNA sequencing, I think there's really an important place for culture-based diagnostics, particularly because a lot of things like, you imagine all of the information we have about the genomes of existing NTM comes from isolation and culture and whole genome sequencing. So if you stop having that capacity, and in fact, almost all of the described mycobacteria were initially isolated in clinical labs. If we switch over to some kind of genomic only workflow, we're going to lose the capacity to understand new and emerging mycobacterial infections. And all of the information that exists about predicting drug resistance from genomic data is predicated on understanding what the phenotype of the organism of the genome you have. And so if we lose that capacity, we are going to lose the capacity to better refine the genomic methods. So I think personally both are required.
All right. It's 12:57. Shall we wrap up now? Okay. So I'd like to thank Luke for presenting. And since you presented with a couple of minutes left, I just wanted to take this opportunity to note that we have a new faculty member here, and Anne-Marie Bourgault is in the room, and it's her last day of work. The first infectious disease specialist in Canada, the head of the LSPQ and the head of labs. So thank you very much, Emily, for many years. Have a good afternoon.